Thanks and good morning. So, pulmonary hypertension. This is an increase in blood pressure in the arteries of the lungs. This requires more work by the right ventricle to pump blood through the lungs, and if uncorrected, can often result in right heart failure. It occurs in around 20% of very low birth weight preterm neonates. It's even higher incidence in those with bronchopulmonary dysplasia. This is a very common chronic lung disease of infancy. Pulmonary hypertension increases morbidity and mortality. There's 50% mortality by 2 years of life and a 4.6-fold higher risk of death in BPD patients with pulmonary hypertension relative to those without. The underlying mechanisms that cause pulmonary hypertension are poorly understood. Early screening methods are unreliable, and there are currently no biomarkers that will predict the onset of pulmonary hypertension later in life. As such, pulmonary hypertension is often not diagnosed until the disease is advanced and associated with severe right ventricular dysfunction. And also because the underlying causes are poorly understood, it's often not clear what is the best way to treat patients, even when they've been diagnosed with pulmonary hypertension. A recent study looked at a correlation between fetal growth restriction and pulmonary hypertension. So it's thought that hypertension occurs during critical stages of lung development, both prenatally and postnatally. In this study, they took a cohort of patients diagnosed with moderate to severe PPD, of which 28% were diagnosed with having pulmonary hypertension at 36 weeks. On the left, you see the bar graph of the patients with pulmonary hypertension, and almost 60% of those had a birth weight below the 25th percentile, relative to those on the right without pulmonary hypertension, were about 20% below the 25th percentile. So these data suggest that fetal growth restriction is a significant predictor of developing pulmonary hypertension later in life. There are several different rodent models of pulmonary hypertension, and newborn rodents in particular of interest because they are born developmentally immature with respect to their lungs, and so that's similar to preterm infants. Neonatal rodents exposed to chronic hyperoxia, which is 75 to 95% oxygen for 14 days, develop pulmonary hypertension and right ventricular hypertrophy. And this model may mimic the hyperoxic exposure in the immature lungs of preterm infants who suddenly have to breathe room air. We're also interested in a rat model of postnatal growth restriction. And here we simply just adjust the litter number. The control group has 10 pups. The nutritionally deprived group has 17 pups. At 14 days, we can see on the left, this is a pup from the control group, and on the right is a nutritionally deprived rat. So because the fetal growth restriction is a predictor of pulmonary hypertension in preterm infants, we hypothesized that the combination of oxygen and nutritional deprivation would exacerbate the effects of oxygen alone. So we simply take groups of either 10 or 17 pups, leave them in room air or in a chamber containing 75% oxygen for 14 days, and then we analyze the hearts, the lungs, and blood. We use right ventricular hypertrophy as a surrogate for pulmonary hypertension by Fulton's index, and this is simply the weight of the right ventricle divided by the weight of the left ventricle and septum. The black bar to the right shows what's been known before, that exposure to oxygen increases right ventricular hypertrophy, but we were really surprised on the left. We found that nutritional deprivation in room air alone also induced right ventricular hypertrophy. And as we hypothesized, the combination of both on the very far bar graph exacerbates the effects of oxygen alone. These changes are associated with a decrease in lung vessel number, increased thickness of the pulmonary arteries, and altered gene expression, all of which indicate pulmonary hypertension, suggesting that nutritional deprivation represents a novel model of pulmonary hypertension. Our overall objectives are to understand the mechanisms that trigger pulmonary hypertension in preterm infants. And the goal of this particular study is to identify candidate biomarkers that will be predictive of pulmonary hypertension and also identify potential therapeutic targets. So in our pilot and feasibility study, we collected lung and plasma from hyperoxia-exposed and nutritionally deprived rats at 14 days. We collected cord blood from a cohort of patients diagnosed with moderate to severe BPD with and without pulmonary hypertension. We analyzed for primary metabolites, complex lipids, and oxylipins, and we're particularly interested in markers of oxidant stress and impaired nitric oxide signaling from evidence within the literature. So a very simplified version of how blood vessels dilates in the endothelium. Endothelial nitric oxide synthase converts L-arginine to L-citrulline and nitric oxide. The nitric oxide then triggers a cascade leading to elevated cyclic GMP in the smooth muscle, which triggers more cascades, ultimately leading to smooth muscle relaxation and pulmonary arterial vasodilation. We looked at levels of arginine, citrulline, and ornithine, and we found that in the nutritionally deprived and oxygen-exposed rats, there was a significant decrease in the eNOS substrate L-arginine, with an even further decrease when oxygen and nutritional deprivation are combined. There's a similar trend with citrulline and ornithine, although a lot of these weren't significant for the N of 4 that we analyzed. In addition, we also found decreases in L-arginine in the lung tissue that we analyzed, and these were associated with a decrease in eNOS expression. So all of these events can potentially impair normal blood vessel dilation, which can, of course, lead to pulmonary hypertension. We tried to detect cyclic GMP, but we couldn't do that in these samples. So looking at the metabolic profile of lung in the hyperoxia-exposed rats, we see several interesting things: altered xanthine oxidase activity, release of oxylipins, acidemia, flux into pentose phosphate pathway, altered redox status. In the plasma, similarly, we see altered redox status, acidemia, altered xanthine oxidase, in addition, altered LOX-CYP activity, and hyperlipidemia. So reactive oxygen species are known to induce pulmonary hypertension via multiple mechanisms, and the data I just presented showing alterations in redox status, xanthine oxidase, LOX, and CYP activities would all suggest increased ROS. In addition, the flux into the pentose phosphate pathway increases NADPH, and NADPH oxidases are a major source of vascular ROS and are associated with pulmonary hypertension. Acidemia is also associated with pulmonary hypertension. 3-hydroxybutyrate is a marker of acidemia, and this is particularly interesting because it's a histone deacetylase inhibitor, and some recent studies have shown that even brief exposures to oxygen can have severe implications later in life, for example, being more susceptible to cancers. Here we see the nutritionally deprived lungs, and we were a little surprised not to see more changes, but we do see some acidemia again, and alterations in nitric oxide production, release of diols. In the plasma, as well as acidemia and increased 3-hydroxybutyrate, there's also some evidence of increased ROS production, altered soluble epoxide hydrolase activity, and dyslipidemia. So it seems that the two models have slightly different metabolic profiles, but the commonalities include acidemia, increased 3-hydroxybutyrate, dyslipidemia, altered soluble epoxide hydrolase activity, and this is particularly interesting because SEH inhibitors have been shown to attenuate pulmonary hypertension in other models of hypertension. The main difference seems to be that oxygen exposure increases oxidative stress, which is perhaps what we would predict. Going to the cord blood, as I said, this is from a cohort of preterm infants diagnosed with moderate to severe BPD. 28% had pulmonary hypertension at 36 weeks. We analyzed 21 samples of each, and the table on the right, these seem to be fairly well matched for gestational age, ethnicity, and gender. One of the striking things we found was that there is dyslipidemia in the preterm infants with pulmonary hypertension, decreases in glucosylceramides, phosphatidylcholines, and sphingomyelins. There also seems to be an increase in free fatty acids, which also could be a potential diagnostic marker as an indicator for pulmonary hypertension. We also see an increase in circulating epoxides and diols in the pulmonary hypertension samples, indicative of soluble epoxide hydrolase activity and also lipoxygenase activity. So in summary, preterm infants with pulmonary hypertension exhibit dyslipidemia. Phosphatidylcholines are particularly interesting because they make up a major component of lung surfactant. The elevated levels of circulating epoxides and diols suggest elevated lipoxygenase and soluble epoxy hydrolase activity, and so these may all provide candidate molecules for therapeutic intervention and diagnostic biomarkers. And in summary of our extremely interesting and fruitful West Coast Metabolomics Center pilot study, we confirmed our hypothesis of impaired nitric oxide signaling and increased oxidative stress in rat models. We've generated novel data, potentially identifying biomarkers and therapeutic targets. We further validated our model of nutritional deprivation as a model of pulmonary hypertension. And interestingly, we've identified a lot of future potential studies, which include potentially arginine supplementation, soluble epoxy hydrolase inhibition, xanthine oxidase inhibition. Some of the data suggest elevated inflammation, which is known to be associated with hypertension, potentially gene imprinting with 3-hydroxybutyrate levels, and also we would like to analyze surfactant composition. In terms of the metabolomic analysis, future work includes looking at a slightly different model that we have where we reduce the number of pups to 5, and we find that that is protective of hyperoxia-induced right ventricular hypertrophy. So we'd really like to know what's going on in those pups. As more patients become enrolled in the cord blood cohort, we'd like to analyze those and perhaps even begin to identify long-term associations with even adult hypertension. The right ventricles and potentially even the liver is also of interest for future studies. So I'd just like to acknowledge everyone involved in this, and in particular, Johannes and Mike for their analysis and also their very interesting discussions. Thank you.
Do you think that diagnostic biomarkers might be found in the mother's blood as well prior to birth, or only the infant?
No, absolutely, that's certainly a possibility. There are all sorts of different potential etiologies, including whether the mom is a smoker, for example. So certainly lots of potential there too.
Thank you. Other questions? Yeah.
So can you elaborate more about gene imprinting you are planning? And other thing, on one slide you mentioned about HDAC inhibitor activity. So that was total activity or for acetylated form or mono or trimethylated?
To answer your second question first, I forget what the inhibitor was. I'd have to go back and look at that. The gene imprinting, so for example, there's studies in nematodes that show that if you're exposed to oxygen for a very brief period of time, it significantly decreases their lifespan. And children kids with BPD and hypertension, unfortunately, are often ventilated with high levels of oxygen just to increase the oxygen in their blood. And it's becoming clear now that some of these kids are actually more susceptible to certain cancers later on in life. And so that's why we think there's some gene imprinting. What we'd like to see is whether we take our oxygen-exposed rats out, crossbreed them, see if there's evidence of those having evidence of pulmonary hypertension in the next generation. As to what the imprinting is caused by, we don't really know yet.
The premature-born infants are on total parenteral nutrition, so they will receive lipids. So I was wondering, since you made a statement that you saw dyslipidemia, how did you control for that?
That is a really good question. At this stage, I'm not really sure how we could control for that.
One more question?
Hi, I'm just curious that you're seeing sort of a negative impact of nutritional lack of nutrition, whereas in adults, it's shown that calorie restriction increases lifespan and reduces the effect of all these diseases. So have you looked at, say, non-nutritionally deprived adults and normal adults and looked for a difference between the children and the adults in that situation to see if there's some kind of switch in later life?
Right, that again is a good question too. We haven't done that, but as I mentioned with looking at the imprinting studies, we are starting to analyze the adults now, so that will be certainly something to look at in future.